Welcome everyone. So I'm Katia Boniface from the ESDR, and together with Hiroyuki Murota from JSID, we have the pleasure to chair this ISID Flavor webinar on genetic architecture of skin diseases. And we would like to thank Takeda, who is supporting this webinar. So today we have two internationally renowned speakers, so Yukinori Okada and Francesca Capon. So I will start by introducing Professor Francesca Capon, who obtained her medical and PhD degrees in Italy. So afterwards, Francesca, you moved to UK in the University of Lancaster, where you became a professor in immunogenomics. And so 2 years ago, you were appointed as chair of genetic medicine at the University of British Columbia in Vancouver. So you are well known for your work in the field of immunogenetics, and in particular by integrating computational and experimental approaches in immune skin inflammatory disease. So thank you for accepting our invitation, and we are really looking forward to your talk.
Thank you very much for the introduction. Okay, so thank you again for the introduction. Good afternoon, everybody. What I would like to do in the next 10 minutes is tell you a little bit about the work of my lab on pustular form of psoriasis, because I think that's a good example of how genetic studies can inform the treatment of rare skin diseases. So before we start, a very quick recap about pustular form of psoriasis. There are two main forms of the disease. So on the top panel, you can see the presentation of generalized pustular psoriasis, or GPP. This is the acute form of the disease, which presents with widespread pustular rashes that are also accompanied by signs of systemic upset. On the bottom panel, you can see palmoplantar pustulosis, or PPP, and that's the chronic and localized form of the disease where pustular eruptions are seen on the palms and the soles. And almost 15 years ago now, our lab and the group led by Hervé Bachelet in Paris found that generalized pustular psoriasis is associated with recessive mutation in a gene called Al36RN. And we also showed that these mutations caused uncontrolled IL-36 signaling. However, when we looked for Al36RN mutation in cases of palmoplantar pustulosis, we found very few. So early on, genetics was telling us that Al36 signaling is a key driver of generalized pustular psoriasis, but not of palmoplantar pustulosis. And this was borne out by the results of clinical trials. In fact, clinical trials of Al36 blockers in generalized pustular psoriasis showed safety and efficacy, and one Al36 inhibitor, spesolimab, has been approved by regulatory agency and is now in use in the clinic, and a second agent is making its way down the drug development pipeline. On the other hand, trials of Al36 blocker in palmoplantar pustulosis failed to meet their primary endpoint. And so these results really told us that if we wanted to find the therapeutic targets for the treatment of PPP, we needed to go back to the drawing board. And so we did. And we worked in collaboration with a multi-center consortium in the UK and Europe and beyond, and were able to ascertain a rather large patient resource that we systematically, examined the features of palmoplantar pustulosis and generalized pustular psoriasis. And what we found is that PPP differs from generalized pustular psoriasis in terms of its clinical presentation, for example, age of onset, in terms of the risk factor, and as I mentioned earlier, in terms of the prevalence of Al36RN mutation. So this is a distinct skin diseases. And to try and understand a bit more what was driving it, we decided to go for a transcriptomic approach. And we started by doing a very straightforward bulk RNA sequencing experiment where we looked at the genes that were differentially expressed in PPP skin. And what we found is that the genes that are upregulated in PPP skin tend to map to pathways that relate to T cell activation. You can see them listed here with Th2 activation featuring quite prominently. And we investigated this a bit more by carrying out single-cell RNA sequencing in the PBMC of affected individuals. And we again found abnormalities in the T cell compartment and identified this T cell population that expressed marker of Th2 and Th17 cells, and we found that this dual positive T cell population was increased in frequency in cases compared to control. And so this was a bit surprising because Th2 pathway, as we all know, are implicated in diseases such as atopic dermatitis, whereas palmoplantar pustulosis is traditionally classified as a pustular variant of psoriasis, and psoriasis is, of course, a Th17-driven disease. So the involvement of Th17 responses was to a certain extent expected, but not that of Th2 signaling. But then we also have to acknowledge that transcriptomic studies have some limitations. Our was relatively small and therefore particularly vulnerable to patient misdiagnosis. But even beyond that, regardless of size, transcriptomic studies cannot discriminate between alteration in gene expression that are a cause of inflammation or alteration in gene expression that are a consequence of inflammation. And to differentiate between the two, you need a large-scale genetic approach. And so we decided to bite the bullet and carried out the first genome-wide association study of PPP. And so again, we work with the Plum Consortium in the UK and with Biobank in Norway and Finland, and we're able to ascertain almost 1,500 palmoplantar pustulosis cases, which is quite a respectable sample size for such a rare disease. And this is the Manhattan plot of our genome-wide association studies with the association p-value on the left and the chromosome position, on the y-axis and the chromosome position on the x-axis. And as you can see, we found a rather significant association with the IL-4 gene region, and IL-4 is, of course, a master Th2 cytokine. And even beyond that, when we looked at pattern of SNP association across the entire genome in PPP from our GWAS and in atopic dermatitis from published GWAS, we observed a significant genetic correlation between the two diseases. So we now also have genetic evidence for an involvement of Th2 responses in PPP. So this has implications for treatment, and in the last couple of years, 2 studies have been published which used our work as rationale for using Th2 blockers for the treatment of PPP. And with the caveat that come with the analysis of small case series, both studies reported that these agents were efficacious for PPP treatment. And an alternative view is that if we want to inhibit both Th2 responses, but also the Th17 responses that were implicated by our single-cell RNA sequencing studies and by subsequent transcriptomic studies, we need to use agents that can block both pathways. And this is where JAK inhibitors are quite an attractive option. And so, for example, our collaborator, David Gleason at St. John's Institute of Dermatology, has just completed a feasibility trial of JAK inhibitor for the treatment of PPP. And then a final word about smoking. Cigarette smoking is a very well characterized risk factor for palmoplantar pustulosis. And so we decided to have a closer look at how it might influence disease pathway. And we use this approach called Mendelian randomization, which is a genetic technique that allows you to demonstrate the causal influence of an environmental exposure on an health outcome. And here we use Mendelian randomization to show that cigarette smoking causally influence the blood levels of several inflammatory molecules. And then that one of these inflammatory molecules that are influenced by smoking can causally influence PPP risk. And this molecule that links, if you want, the effect of cigarette smoking to PPP risk is in our analysis OX40. And that is a very important protein for the activation of T cells. So I hope I've convinced you that genetic studies have shown that palmoplantar pustulosis is a distinct clinical entity from generalized pustular psoriasis. It is mediated by the activation of Th2 and to a certain extent Th17 pathway. And according to our Mendelian randomization studies and independent work carried out by others based on spatial transcriptomics, OX40 could be an important disease mediator. And interestingly, OX40 is required for both Th2 and Th17 cell activation, and it is a druggable target. Moving forward, I think we can learn more from genetic studies. We would like to do possibly larger GWASs, working in collaboration with other biobanks. And we've also started to look at Mendelian forms of the disease. And for example, you can see here a small pedigree where 4 individuals are affected by SAPHO syndrome, which is an autoinflammatory condition where PPP occurs in conjunction with bone inflammation. And we have completed the whole exome sequencing of this pedigree, are looking at candidate gene studies. And in fact, if anyone has other SAPHO cases and would be interested in collaborating with us, we would be very happy in hearing from you. And so my final slide is just to acknowledge the consortia and the funders that have supported us, the students that did all the work, and the clinician that made it possible. Thank you very much for your attention.
Hello, everyone. So I am Hiroyuki Murota from JSID. I'm very excited to co-chair today's ESDR Kitchen and ISID Flavors episode. So now it is my great pleasure to introduce Professor Yukinori Okada. Professor Okada is a physician scientist and a leading expert in statistical genetics and genome informatics in Japan. So he is now professor of the genome informatics at the University of Tokyo. He also holds positions at Osaka University and RIKEN. His research uses large-scale genomic data to understand the links between the genetic variation and human traits and the diseases. So Professor Okada, thank you very much for your joining today. So now, screen is yours. So please go ahead.
Okay, hello everyone. This is Yukinori from the University of Tokyo. And yes, again, thank you so much for giving me such wonderful opportunity to have talk this. I think it's connection between Japan and European Dermatology Society is very fun. So I'm going to talk about something related to the previous talks of Francesca. So how genetics, human genetics, statistical genetics help us to understand the skin diseases. Yep. Okay. So yes, I first talk about my slide already this slide. So maybe human genome sequence. This is the blueprint of humans, which define ourselves, diversity, and also biology. So if you can fully understand the meaning of your genome sequence, you can reconstruct yourself and everything. So because of the development of the main technologies, we are now very nice era to go tip to the iceberg to the bottom to the iceberg. And we now get so many layers with the omics layers. So population scale, individual scale, and single cell scale regions omics. So population scale, most of the case GBUS, but we can now get many layers, and projecting each layer into different layer help us to understand disease biology. And human genetics, this is a very nice tool to connect these different types of layers. And yes, some of the slides in Francesca. So yes, we are now using so many huge big data. This is the UK Biobank, which have released 500,000 individuals genome phenotype data. But now I think there is much for other global biobanks with 100,000 individuals. So if you do not have any sample, you can start your study from millions of individuals. It's a very wonderful resource. And I think most people already done GBA, something like to identify disease gene, but there's so many things which we haven't explored. So this is a very interesting resource. And I have been working on some of biobanks in Japan. For example, Biobank Japan. This is the very one of the successful disease hospital biobank in Europeans. Previous several years before we conduct GBAS, more than 200 phenotypes, which cross population, UK biobank, which provide us tons of the genotype phenotype associations. But the most important is the data should be publicly available. So we construct the database named field.jp. So you can go to this database and get all the data. For example, okay, let me see. You can put the name of the genes, disease, variants of your interest. When you put atopic dermatitis, it's a kind of here GBUS. And when you put download summary statistics button, you can get everything summarized. Yes, it's a kind of typical allergy disease GBUS result. Many interleukin-related genes. And yes, here's a strong peak at the chromosome 6 MHC region. So first part of my talk, I'm going to sum about this MHC region stuff. So yes, this is MHC region, but you may know that this includes multiple classical and non-classical, or class 1 and class 2 HLA genes, which have wide range of phenotypic impact on immune-related or cancers, psychiatric disease, so many stuff. And HLA genotyping is still costly, but using the genotype data and machine learning methods and population-specific reference panel, you can in silico impute the genotypes to HLA. So you can now do the HLA-based association study if you have the GBUS data. So I learned this method more than 10 years before when I was postdoc at the United States. And my first MSc imputation analysis was the psoriasis. Yes, I occasionally get the collaboration with JTL with Michigan. And yes, I applied HLA MSc imputation to 220,000 psoriasis cases. And what we found is, yes, HLA-C defined the risk of psoriasis. But once you got the psoriasis, whether you got the arthritis or not, it's defined by HLA-B, not C but B. So different HLA genes and alleles have different affects the risk of the disease and prognosis of diseases. And then came back to Japan. I started to do HLA analysis of psoriasis in Japanese population. So this is interesting that C0602 is the strongest risk in psoriasis, but this is almost rare in Japanese. So the psoriasis patients in Japan is mostly C060602 negative type. We found that in Japanese population, or I think in others, Asian as well. So HLA and DQ, A amino acid polymorphism has much more strong impact on psoriasis risk. But what happened? So people always say HLA is a risk or not, but what is they doing functionally? So they are affecting how to use TCR, CDR3 repertoire sequences. So when you do the single cell sequencing, especially 5' time, you can get the TCR sequence of each T cell. And maybe check the association. HLA has some preference to get which VDJ genes of the TCR. So class 1 HLA association affect the VDJ and CD4 T cell. And class 2 HLA affect the VDJ sequence CD8 T cell. And the usage is depending on disease state. It means under the different stimulation. Anyway, your HA arrays define which BJ and repertoire you said, and then this affect the risk of the diseases. Okay, maybe my next talk is about more genome-wide stuff. So whole genome sequence, rare variant, and structure variant analysis or psoriasis. So I started this more than 10 years before, especially the strong collaboration with Professor Murota and the Nagoya City University. And we concentrated on the initial whole genome sequence-based psoriasis GBAS. And I'm going to show some of the results on this study. So we conducted whole genome sequence-based GBAS of psoriasis. The point is this is whole genome sequence. So we can do rare variant association study and also gene-based rare variant test. So when we do common single nucleotide variant GBAS is, here it's a kind of typical GBAS result, MSC, TNF1, and interleukin. But when we do gene-based rare variant test, we found another gene, something like interleukin-16 and circum. It's surprising because circum is the name of the gene which we newly identified. Never GBAS common variants pointed to these risk genes, so which we are wondering whether this is a true association or not. So we constructed the knockout mice of the circum gene, and yes, the circum is specifically expressed in the specific type of skin fibroblast. So SARP2 type fibroblast, and we confirmed the distribution using spectromics. And when we constructed knockout mice of the circum, we strongly showed that imiquimod-induced psoriasis mouse model aggregates and dermatitis. It also led to the T-cell adhesion and something stuff. So yeah, yes, it's social circum that truly causes gene for psoriasis. So I want to say that rare variant association study for genome sequence can give the new genes which haven't been implicated in the GBASs. And we also conduct the fine mapping of the GBAS signals by using short and long read from sequence data. For example, we found that GBAS heads in the interferon LR1 gene is a kind of famous immune gene. We found that here's a 3.3 kilobase, it means 3,000 base pair deletion structure variants, which disrupts the enhancer region of this immunity gene, and this causes the psoriasis risk. So yes, I want to say GBAS is very strong, but sometimes they miss risk gene, which irreverent can help us. Whereas GBAS is very interesting, but they sometimes miss the truly causal variant, something like structure variants. So in a combination of GBAS and rare variant for sequence study will help us to more pinpoint the psoriasis or other skin disease risk genes. Okay, this study has been helped by so many collaborators, and I would like to thank you to all of my collaborators and also my colleagues in Japan Society Investigating Dermatology. Okay, thank you for listening.
Thank you very much, Professor Okada and Professor Francesca Capon, for these two very nice talks. So now you can ask your question to the speakers, either on the chat or in the Q&A. So we had a question from Michel Simon, so in France, to Francesca, who is asking, how do you explain that the lesions in PPP are mainly observed in palmar and plantar skin and are not on the whole body?
Yes, that's a very good question. Thank you. Well, one of the possibilities that, of course, palmar and plantar skin is different. It's from the rest of the skin of the body. It's different at the histological level, as we all know. And there have been studies showing that it is different in terms of there are some difference in terms of the resident T cells. And then there is a theory that part of a cell type that is particularly, a structure that is particularly important is the acrosyringium, which is basically the duct of the sweat gland that are found more densely on palmar, on the palms than elsewhere in the body. So it's probably has got to do with the histology of this skin.
While we still wait, so I had a question about like cross-ethnic diversity, because so it's maybe for Professor Okada, but maybe also for you, Professor Capon. So how crucial is it that we have to perform cross-ethnic genomic data to really identify which biologic drugs we should use and could work like equitably across all patients? So I don't know if we can like take the advantage of all these genomics data across like all different ethnic to try to adapt that.
Well, if I can start, well, we should, obviously, for reasons of equity, so that everybody, that the results of these genetic studies can be equitably shared, of course. But also there is power in, and I'm sure Professor Okada can further comment on that, but there is power in harnessing the fact that linkage disequilibrium, the way linkage disequilibrium is conserved varies across population, and therefore by comparing and contrasting results obtained across population, we also increase the resolution of our studies. But I'm sure that Professor can comment further on this.
Yeah, yes, I fully agree. So the cross-population study definitely increased the power of the discovery and also help us to find out the causal variant because each population different LD. But I think we finally found that somehow disease biology or fraction of the disease subtypes slightly differ across population. So we can stratify the patient into several pathway-based diseases and based on genetics, and we always find that fraction different across population. So when we compare the genetics of different population, we finally get the novel biologies. And finally, we also need to return the GBUS result or prediction model to each population. In this case, the GBUS from diverse populations should be really necessary to make a nice training model. So anyway, yeah, cross-population studies is definitely important.
So we have a question from Aldona Pietrax. So do you mean that palms and soles can have another type of receptors for resident cells? I think it. So Aldona Pietrax is asking, do you mean that palms and soles can have another type of receptor for resident cells?
Well, not exactly another type of receptors, but so there have been studies of the distribution of skin resident cells, and there seems there is some data to indicate that there may be more Th17 cells resident in the palms than you would see in other part of the body, and the microbiome, of course, is different on the palms because they are more exposed to microbiome than other areas of the skin. So there are different in terms of skin structure and different in terms of microbiome and difference in terms of skin resident T cells, if that makes sense.
So may I have a questions? So Francesca, thank you very much for your excellent talk. And so your work beautifully connects genetic risk with disease biology. And as a next step, do you think integrating genomics with epigenomics will help us understand how genetic risk become active in specific skin or immune cells?
Sorry, interrogating what? Epigenetics, you said?
Yeah, genomics,
Yes.
integrating genomics with epigenomics.
Yes, I think that would be really, really interesting thing to do, especially as, for example, in palmoplantar pustulosis, cigarette smoking plays such an important role. I mean, almost 90, in some studies, up to 90% of patients with palmoplantar pustulosis are smokers. Yes. So clearly there must be some epigenetic changes. So that is, that is an experiment that we would be really interested in carrying out.
Thank you. And also I have a question to the Yukinori. So your work shows that integrating different types of data can reveal new biological insights. And to make this approach even more powerful, what kind of data should we collect and organize better in the future?
I think the last several years, the researchers are massively trying to collect single cells of skin or single nucleus. But I think now people are moving to the spatial omics, spatial transcriptomics, or spatial proteomics. The technology still hasn't been fixed, but I think the current spatial data is very nice. For example, to project to give us results to understand the 3D accumulation of the risk. So anyway, yeah, spatial transfer is still hard for skin, but I think it's very promising technologies.
Okay. Thank you.
And maybe one last question, so to both of you actually about JAK inhibitors, because that's true that now when we want to target several pathways, so that's what we are saying, that if we use JAK inhibitors, we will like target both like Th1, Th2, Th17 pathway. But if we take the example of pustular psoriasis, which where like IL-36, for example, has been shown to be really involved, if I'm correct. So it's not like a JAK-mediated signaling pathway cytokine. So how do you boost for this some cytokines like the IL-1 beta, IL-17 or IL-36 pathway, which are kind of independent of JAK signaling? So how do you think that it, like JAK inhibitors, have their place in this case?
So that's, I think, where genetics is really helpful, because genetic studies have shown that, for example, generalized pustular psoriasis, the really rare and severe form of the disease, is an IL-36-driven disease. And there, IL-36 inhibitors have really shown their efficacy. Whereas palmoplantar pustulosis, where there seems to be this widespread activation of T cell response along multiple pathways, that is more difficult to treat with biologics that only hit a single axis. And so that is where JAK inhibitors can really come into their own, with some caveats, as we know there are some safety concerns around JAK inhibitors, and with the risk of major cardiovascular events, and the people who suffer from PPP tend to be smokers. So there is a little bit of caution there, but in terms of the scientific rationale, the mechanistic rationale for using those agents in PPP, I would argue there certainly is one. That would be my view.
Yeah, maybe I have a quick comment. Yeah, I fully agree with Francesca's opinion. So anyway, human genetics is now trying to stratify the patient into several pathways highlighted as subgroups, and definitely the biology, immunology, and treatment response is strongly different across these. So maybe how to utilize genetics and current immune phenotypes to subtype the patient is very important to maximize the value of the drug. And I think it's also true for JAK. Yeah, thank you.
Thanks again to both of you for these two wonderful talks. And now the webinar is coming to its end. So as a reminder, so the webinar was recorded, and so it will be available in a few days on the ESDR Kitchen webcast on the YouTube channel. And so the next webinar in June will be a sweet and sour episode on skin resident immune regulation. So with Dan Kaplan and Claire Bennett. And so as a reminder, so in September, so from 9 to 12, we will have our annual ESDR meeting, which will be held in Heidelberg, Germany this year. So you can still submit your abstract until July 12. And so we have a wonderful program with a very nice speaker coming all across Europe and even from further countries. So we hope that you will all be able to attend in September. So thank you very much to all, and so looking forward to the next webinar in June. And thanks again, Francesca and Professor Okada. Goodbye.
Goodbye. Thank you.
Bye.
Bye.
Thank you.
See you.